Hi, my name is Nico Scigliano, and this is my presentation on how close are we to modifying the human genome. So first off, I want to talk about the technology that's being used to modify the human genome, and this technology is called CRISPR, or Cas9. Cas9 is the protein of this technology. So CRISPR-Cas9 is a DNA modifying tool that was discovered from research being done on bacteria. So basically, this was discovered in around 1980, I think it was, and it was extracted from bacteria, and research really didn't know what it was capable of. So really, nothing was done with it until around 2010, where research on CRISPR kind of skyrocketed. So basically, how this works is we're gonna have our CRISPR-Cas9 protein right here, and lab researchers are going to make this thing called guide RNA or lab synthesized RNA. And what this is going to do is this guide RNA is going to take our Cas9 protein down to a specific target DNA because this RNA is specifically made for this section of DNA. And what it's going to do is it's going to attach to our gene, and it's going to cut our gene using these clippers right here, and it's going to allow our gene to unwind. And so what's going to happen is this guide RNA is going to come in and it's going to attach to our DNA, and it's going to shut off the genes down this way of the molecule. So that means we won't get expression of these genes. Most of the time this is done for some sort of bad gene that we don't want, right? But we can also use this. There's also a CRISPR-Cas9 protein that doesn't cut, and all it's going to do is unwind our DNA, and this is going to attach, and it's actually going to let us totally change the gene versus actually stopping the transcription of a certain gene. So we can actually change a bad gene into a good gene, or change an okay gene into a great gene. Another thing that we can do with this technology is we can add a fluorescent onto our Cas9 protein, and this Cas9 protein is going to attach to our DNA, and it's going to allow us to see genes transcription of certain bodily functions. So we can tell on a chromosome where that DNA transcribed, where that DNA is located for certain bodily functions. Another thing this can do is we can also increase transcription. So if we want more transcription of a certain amino acid to make a protein, we'll take CRISPR and add new genes that increase the transcription rate. So my first argument is we are close to editing the human genome. So recently there has been a lot of research backing this, and it makes people believe that we are close to editing the human genome because there's such a push for this research right now. The first research I want to talk about is a researcher at Oregon University of Health Sciences. His name is Shavkat Matalipov. I think I'm saying that right. He actually successfully genetically modified human embryos using CRISPR. Now, these were single cell embryos, and he was actually able to use CRISPR to take a certain synthesized DNA or synthesized RNA and edit the DNA within a chromosome of a human embryo. Now, this has a lot of moral complications, but we'll talk about that later in this presentation. Also, Chinese researchers have successfully been able to edit human white blood cells, and they took these white blood cells and implemented them back into the human, and there has been no major consequences of doing this. Now, they haven't released a scientific journal since they did this, so they haven't released all of the research that they've done, but they just said they safely were able to edit these T cells and put them back into a human. These recent discoveries between the U.S. and China have kind of elevated the competition, so it's kind of like the space race with the U.S. and Russia, where it is a race to the moon. This is kind of the race to being able to genetically edit human beings, and this is going on between the U.S. and China right now. So there's huge amounts of research being done in both of our countries. The second argument that I want to talk about is we are not close to editing the human genome. So researchers aren't really sure on how editing the genome is going to affect the human long term. So changing one gene in our body could result in fatalities or even worse complications than that were originally trying to be treated. So in a lot of the research being done, especially with the human embryos, none of these embryos were actually developed and actually born or anything like that. So we don't know how the human embryo would have been affected as it developed with the changing of his genes. So we have really no idea on how to do this. And because we would have to actually test and do clinical trials on human embryos and babies and stuff like that with this technology, this kind of brings in complications with the FDA and actually approving the research for this to do clinical trials. The third argument is it morally right to alter the human genome? So this is definitely the biggest controversy, and the controversy that we could talk about a lot, or the argument that we could argue about a lot. So this technology could potentially save millions of lives. It could get rid of birth defects. It could potentially help in curing cancer and heart disease and multiple things like that. So we could save millions of lives, but an argument to that is that this technology may only be available to those who can afford it. So it may be it originally will be very expensive. So basically, it'll be a way for the rich to get richer in a way. And also, this is a technology that can be used in the wrong ways. For example, it could be used to make super armies and stuff like that. Because if you can edit one gene, you can edit the gene for height, weight, muscle mass, stuff like that. So, and also we could be playing with laws of nature a little bit too much, because with saving some, I mean, this sounds terrible, but the more lives you save, the more the population increases and the resources decrease. So if we don't have enough resources to take care of all of these people on the planet because we're saving so many people and nobody's really dying, that presents a problem in itself. Who does this issue apply to? This issue applies to everyone sitting in the room. It's something that could affect us in the near future or in our lifetime. There's a possibility. It's a technology that could benefit, would definitely benefit our world in a number of ways, but as long as it's used in the right way. If it's not used in the right way, then we could potentially see drastic problems with it. And that is the end of my presentation. Thank you for watching.